We now conclude our tour of lipids by examining the fat-soluble vitamins. So vitamins is the general term used for relatively small organic molecules that are essential for life and must be obtained from food. Initially, people thought that vitamins were all amines, so the term vitamin actually comes from vital amine, but we now know that many of them are not amines, so that's truly a historic term. But what we do know about vitamins is that many of them are what are called coenzymes, which we will have more to say in a subsequent unit of this course. But coenzymes are molecules that are required for an enzyme to function, but are not the actual substrate for that enzyme. The enzyme does not catalyze a reaction on the coenzyme. So vitamins have been divided into water-soluble and fat-soluble. And since the fat-soluble vitamins, which are vitamins A, D, E, and K, are lipids, I'm going to talk about those now in this unit, and I will talk about the water-soluble vitamins in a later unit. So vitamin A is involved in the chemistry of vision. So the structure of vitamin A is shown on the right side of your screen. It is also called retinol. And if you count the carbons, there are actually 20 of them. And by this point, you should actually be able to recognize vitamin A as being a diterpene with the methyl groups and the branching that you need for a terpene structure. So vitamin A itself is isomerized and oxidized to a molecule called 11-cis-retinal. So vitamin A is retinol, has an OH at the end of it. Retinal has an aldehyde at the end of it. And then one of the double bonds gets isomerized to the cis position. This is, of course, done enzymatically in the body. So once you have 11-cis-retinal, it actually binds to a protein that is called opsin. And this protein is found in the rod cells of your eyes. Okay, so in order for you to be able to see in your eyes, you have these receptors called rods and cones. And the rods are actually the ones that respond the most to light, and the cones respond more to color. So the combination of the protein opsin with 11-cis-retinal forms a molecule called rhodopsin. And you can see what the shape of rhodopsin is on your slide. So upon exposure to light, the cis double bond in rhodopsin isomerizes. It's a photoisomerization to the trans double bond. And as you can see from the way I've drawn it, but this is actually true in real life, this cis-trans isomerization causes a significant change in rhodopsin's shape. Okay, and you've seen this before, the shape of cis versus trans fatty acids and the effect that that has on things like melting points. So this change in rhodopsin's shape from cis to trans, from bent to linear, if you prefer, initiates a cascade of enzymatic reactions in the body that ultimately cause the release of calcium ions. Okay, so cascades of reactions frequently involve the release of calcium ions. And what actually happens is these calcium ions, because you get so many of them, they block sodium channels, which are simply protein pores through which sodium, being a charged species, can pass in and out of cells. Sodium, being Na+ being charged, cannot easily diffuse across the lipid membrane of a cell. So they have to pass in and out of the cell through these protein channels. So in the optic nerve cells, there are actually always sodium ions passing in and out. That's called the dark current. But when the sodium channels are blocked by these calcium ions, the flow of sodium in and out stops. And that actually results in a nerve impulse that is sent to the brain. And all of this happens in response to the light photoisomerizing the cis form into the trans form. This is such an efficient process that the absorption of even a few photons is sufficient to transmit the nerve impulse to the brain that responds to light. So that's why it is possible for humans to still see in a fairly dark environment, and of course there are animals that have even better night vision than humans do. Okay, so the next vitamin I want to talk about is vitamin D. I'm going to talk specifically about vitamin D3, although vitamin D2 has a very similar structure. It just has one additional double bond. Okay, so the structure of vitamin D3 is indicated on the slide. That is not the way that most textbooks draw vitamin D3, but I have drawn it that way for a purpose. And of course, the purpose is to show that vitamin D3 is biosynthesized in a couple of steps from the steroid 7-dehydrocholesterol through the cleavage of the B ring. So the first reaction in this sequence, we're not covering it in great detail, but the first reaction in this sequence also requires UV light. So one of the things you should be able to see is that UV light interacts with conjugated double bonds. So both vitamin A and the 7-dehydrocholesterol have conjugated double bonds, and that should not surprise you at all, given the fact that we have seen that these molecules absorb UV light through their HOMO to LUMO transitions. And the most common place that UV light comes from, of course, that people can be exposed to is sunlight. So this 7-dehydrocholesterol is actually naturally found in milk. And when you buy vitamin D milk in the store, that is actually milk that has been irradiated with UV light to convert the 7-dehydrocholesterol into vitamin D3. So you don't actually add it to the milk, you just shine ultraviolet light on the milk. Now, you don't get the ultraviolet light that you need to make vitamin D3 if you insist on putting sunscreen on every time you can even remotely be exposed to the sun. Obviously, too much ultraviolet light is bad for you, but some amount of it is necessary in order to make vitamin D3. So excessive use of sunscreens has actually led, some people say, to vitamin D3 deficiency in 50 to 75% of the US population. And so possible clinical manifestations of vitamin D deficiency are rickets. That's more associated with vitamin D2 deficiency, but everything I'm saying about D3 is actually also true for D2. But vitamin D deficiency is possibly associated with cardiovascular disease, diabetes, hypertension, and more recently, depression. So there is some actual biochemical evidence that the sunlight that is necessary to make vitamin D3 puts you into a better mood, and that's why people who live in climates that don't get very much sunlight often suffer from depression, or at least one of the reasons, not the only reason. Okay, so that's vitamin D3. The other two I want to talk about, and I'm going to be much briefer about these, are vitamin E and vitamin K. And if you look at these two structures, they actually also look pretty similar, and they both have diterpene chains coming off of an aromatic ring. So in the case of vitamin E, the aromatic ring is a simple benzene ring, monocyclic. In the case of vitamin K, the aromatic ring is what's called a quinone. So it's a bicyclic aromatic ring that has two ketones on it. And we're going to have more to say about quinones in a later unit of this course also. Okay, so the biological function of vitamin E is that it is a fat-soluble lipid antioxidant that traps radicals that are formed in cell membranes. Okay, so we already saw in our prostaglandin synthesis that radicals are a natural part of biological processes, But the problem, and we'll talk about this a little bit also in a later unit, is an excess of radicals in your body can cause all kinds of problems. So, and those problems are known as oxidative damage or oxidative stress, and also primarily lead to the aging process. So you have molecules in your body, vitamin C and vitamin E, that actually can react with radicals. They get destroyed by the radicals, but they keep the radicals from carrying on other molecular mayhem. And vitamin C is water soluble, so it takes care of radicals in aqueous environments, and vitamin E is fat soluble. It takes care of radicals in the lipid environment of cell membranes. Vitamin K is involved in blood clotting. So the vitamin itself, the quinone, is actually reduced to a diol, and that diol form is actually the active coenzyme for an enzyme that catalyzes the carboxylation of a glutamate residue on a protein that is involved in the first step of blood clotting. So there are actually several glutamates on this protein, and those glutamates get carboxylated at the position shown, so you add another carboxyl group, and once that happens, more molecular recognition happens, other enzymes and factors bind, and that leads down the cascade of forming a protein called thrombin that is involved in blood clotting. So obviously blood clotting is important because when you cut yourself, you do want the blood to be able to clot, but there are also nasty things that happen as a result of blood clotting when we don't want it to clot, which are, of course, heart attacks and strokes. Okay, so hopefully you have seen in the course of these lessons a little bit of an overview of lipids. So the different kinds of lipids we have, we've now covered all of them, and a little bit about their structure, their chemical reactivity, and also about their biological function. So moving forward from lipids, the other major class of biomolecules are, of course, the amino acids and the proteins. But before we get into actually being able to talk about amino acids and proteins, there's a little bit more that I want to tell you about amines themselves. And that will be the first couple of lessons of the next unit.